This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode number 283, recorded on March 1st, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michelle Swanson.
Hello. It is March. Hallelujah.
What? Why? You didn't like February?
We have bright, sunny skies. It's going to be 50 degrees here today. Feels very different from the ice storms that we were plagued with.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
You got 80-degree weather?
We do indeed.
Oh, my gosh.
It was Chamber of Commerce weather yesterday. The students were all out in shorts and flip-flops running around on the grass. Grass is not yet greening up, but the pollen is causing my nose to run.
Oh,
like
my
a
goodness.
drain. Spring has sprung.
Better allergies than frostbite.
Yeah.
Absolutely.
All right. Well, the weather's good in some parts of the U.S., and we have good science for you today, as usual. No matter what the weather, right, we bring you good science.
Absolutely.
We're going to start with a snippet from Michael.
Well, this paper is entitled The Interconnections of Pseudomonas aeruginosa quorum-sensing Systems in Intestinal Permeability and Inflammation. Paper was authored by Singh, Almpenny, Wheeler, and Rami, and they are located in the departments of surgery and microbiology at the Harvard Medical School and the Massachusetts General Hospital and the Shriners Hospital for Children, all of which are located in Boston, Massachusetts. Today's paper was published in MBio, so it's open access, and it's not a full paper. It's called an observation, and I selected it to indulge my fascination a bit more about quorum sensing. I find quorum sensing just absolutely magic, and the magic of quorum sensing comes from its fascinating molecular circuitry that has evolved to respond to concentrations of small molecules or quorum factors exported freely into the surrounding environment that upon their passive diffusion and then encountering either the same species that donated these quorum molecules to the environment or even other microbes receptive and able to interpret the language inferred by the quorum factor results then in the activation of a formerly inactive transcription factor that then results in the transcription of a variety of genes that ultimately do a variety of things, but mostly it leads to fitness advantages for the microbe. And that's basically how we're getting into quorum sensing. So today's paper describes how Pseudomonas aeruginosa, which is a common multi-drug resistant gram-negative bacterial pathogen, can seriously threaten critically ill in immunocompromised patients. We'll learn from that upon establishing itself in the intestine of its host, this pathogen is associated with significantly higher mortality rates, begging the question, how can this microbe be doing this? Well, you guessed part of its ability to cause disease or virulence involves quorum sensing from the title, which is nothing more than a highly conserved form of microbial communications based on this production of these small molecules and then sensing the concentration of the secreted signaling molecules that then results in the novel expression of certain gene products that then alter the phenotype of the microbe in the environment that it finds itself. And the first thing that the authors do is acquaint us with the observation that Pseudomonas aeruginosa has this complex, interconnected series of quorum sensing systems. It actually has 3 that we know of, controlling the expression of multiple virulence factors from three separate and distinct genetic regulatory circuits. Our understanding of quorum sensing has progressed remarkably from those first reports well over 20 years ago from the laboratories of folks like Barbara Glusky and Pete Greenberg, and more recently, folks like Bonnie Bassler at Princeton. And it's continued to blossom in other laboratories, now contributing to our understanding of this complex communication in microbial systems employed by microbes to literally give them a leg up in the niche that they find themselves in. We're going to address or move our understanding today from in vitro to in vivo, where we're going to look at the consequences for Pseudomonas aeruginosa when it finds itself in an animal host, and then how these interconnected and distinct quorum sensing systems translate in the host environment during an infection. We already know that when Pseudomonas aeruginosa infects our GI tracts, that these interconnected quorum sensing systems control multiple acute and chronic functions that contribute to the severe systemic infections that Pseudomonas aeruginosa can cause. And the authors offer over 15 distinct references in support of this statement. Additionally, GI infections of Pseudomonas are often associated with disrupted intestinal integrity, which may then lead to gut-derived sepsis. And we all know that sepsis is a bad time in any host because it unleashes the immune system, and we have all sorts of bad consequences, often which are ascribed with higher mortality rates. Thus, Pseudomonas wound infections and the resulting intestinal barrier dysfunction are important clinical considerations. And you will note that in their studies, they use as if you will, a control, a burn. And they actually will burn 30% of the total body mass in their animal model to effectively stimulate the inflammatory system independent of the infection. So it's an interesting control that they add to their methodology. And one last piece of information in their rationale of why do this study is that in our developing an understanding of the relationships between these three distinct quorum sensing systems, we may then be able to design successful antivirulent strategies that will help us treat this often pan-resistant gram-negative rod. And then one final piece of background, the role that quorum sensing serves in the development of virulence is that often one of the global regulatory transcription factors, the first one that I learned about when I was beginning my understanding of quorum sensing, the LAS-R gene regulator, often accumulates specific mutations during the infection of this microorganism in the host. Now, if you jump to panel C, if you're reading along with us, of their second figure, you will get a sense of where their data are going to take us today. Namely, the level of intestinal inflammation and permeability is driven by the MVFR quorum sensing system. And this is the third system, and their data are going to show that this MVFR triggers tight junction disruption and an increase in intestinal permeability indirectly, at least through the Pseudomonas aeruginosa quorum sensing network, which consists of these three interconnected regulatory systems. One's controlled by this MVFR system, the other controlled by LAS-R. And LAS-R and the second system, REL-R, are often intimately linked. In fact, LASR will actually turn on REL-R based on responses from LAS. You get REL turning on. And MVFR is often implicated in the formation of biofilm formation. And so each regulator contributes to intestinal permeability, with their data offering that this global regulator MVFR functions upstream of LASR and REL-R. This was the O. Henry moment, if you will, to me. I always thought LASR started the parade, but in reality, their data are going to offer us that MVFR is actually upstream in terms of functioning, if you will, in the virulence that we're seeing associated with a Pseudomonas infection in the gut of the host. So that's their model. How they arrived at the proposed model is that they used a collection of Pseudomonas aeruginosa quorum-sensing mutants, all targeting the global transcriptional activators that coordinate the quorum-sensing activities. Remember, these activators turn on genes that are then involved in the expression of various traits in the microbe. And the three major quorum-sensing systems have these interconnections and contributions, and they're all interacting to contribute to intestinal inflammation and this barrier function. Once the Pseudomonas gets out of the gut and goes systemic, that's the bad consequence that no infection ever wants to see. So first, the quorum-sensing regulator, MVFR, is involved in systemic consequences in the host, including increased intestinal permeability and inflammation. They already knew, and the senior author of this paper, and I'm going to drop this manuscript into the show notes, has already published that pharmacological inhibition of this regulator significantly alleviates bacterial-induced gut permeability. It decreases tumor necrosis factor alpha levels, which is a measure of inflammation in both serum and the ileum, and it decreases bacterial dissemination and abolishes the production of these MVFR regulated small molecules that are critical for acute and persistent infections. Now, here's where I got really excited, being the old bacterial geneticist, by looking at the role of these other two quorum-sensing regulators, LASR and REL-R, and their impact on the interconnection to this other regulatory system controlled by MVFR in the gut. They could then determine the consequence of dissemination and intestinal permeability and ultimately inflammation. And they did this simply by using double and triple quorum-sensing mutants. These are deletions that they then help to dissect the consequences of these global regulators on the microbe's ultimate phenotype. Now, this paper only has two figures, but they are data-rich and both are very approachable. The first figure addresses the question when the transcriptional regulators are deleted, which will effectively eliminate all the coordinate expression of the downstream virulence factors, if you will, where does Pseudomonas aeruginosa go? And so collectively, the data from the first two panels offered that the double and triple mutants lacking the regulator MVFR were reduced in both the ileum and colon relative to the wild-type microbe. And to give you some frame of reference, look at the first column on the left, which in panel figure 1A, which is the control, and then the third column, which knocks out MVFR MVFR, and you can actually watch what's actually going on. The inflammatory response was measured in the ileum by measuring the TNF-alpha levels. And so they're measuring TNF-alpha, where they found that the response in the ileum, TNF-alpha levels increase when mice were infected with the deletion of the global regulator LASR, the global regulator RELR, and in wild-type strains, whereas the TNF-alpha levels were similar in the controlled, just the straight-up burn alone in the deletion mutant, MVFR, and the double and then the triple mutants in this. Taken together, these data then offer that the genes controlled by this global regulator, MVFR, but not LASR or RELR, are involved in triggering the intestinal inflammatory response, and that this role is independent of the ability of each mutant to disseminate into the ileum, because we saw that LASR and RELR got in there just as well as we saw with the wild-type PA14 microbe. It was also known that the consequence of mucosal inflammation is associated with intestinal permeability. Again, that's why we have the burn, because that's going to cause general inflammation, but we're looking here specifically for intestinal permeability. So in the fourth panel of their first figure, they measured the flow of a dye that normally doesn't get out of the intestine. This dye is fluorescein isothiocyanate, or FITC, and it's coupled to dextran in a size range of 3.5 to 5.5 kilodaltons. So if you have a good and intact epithelial layer, this dye, dextran, is not going to get into systemic circulation. If, however, your gut permeability is breaking down, then the fluorescein labeled dextran will enter systemic circulation, and you just need to bleed the animal and effectively ask the question, does it fluoresce? So if your gut's leaky, you're going to see a fluorescent spike. And here they observe the floor of the FITC dextran from the intestinal lumen to the systemic circulation was not increased significantly in the deletion mutant of MVFR in comparison to burn alone, whereas in wild type and the delta LAS and the delta REL and the double and triple mutants increased intestinal permeability. So they offer that this suggests that the products regulated by LASR and RELR may also contribute in maintaining the intestinal barrier integrity that are downstream of MVFR. So gut integrity is critical to containing the microbes, or simply put, keeping the sterile parts of us sterile. This is principally mediated by a multi-protein tight junction complex, which is a critical component of our intestinal barrier regulation. And the protein that principally facilitates this is called a claudin, which is a tetraspan transmembrane protein that's associated with these tight junctions. So they have antibodies against claudin, and their next experiment was to then determine how the tight junctions were altered in all of these mutant combinations. And here they use CACO2 cells, which is an intestinal cell line, following infection with each quorum-sensing mutant strain. The outcome that they looked for was tight protein junctions, and the claudin 1 is the protein that they're looking for, and they did this by immunofluorescence staining. And what they saw is that while the cells infected with the knockout mutant to MVFR retained staining of the protein. This is the bottom row, third panel, so you can see it lit up just like the no infection control. And the second panel on the bottom portion of figure 2A shows you what Pseudomonas does. It eliminates all that colodulin from the CACO2 cells. So, in other words, they would expect them to get leaky with Pseudomonas aeruginosa, thus facilitating escape from the gut, and Pseudomonas aeruginosa being able to become systemically invasive. So their observation was that the integrity of the tight junction protein colodulin was compromised when cells were infected with wild type, the delta LAS-R mutant, or the delta REL mutant, and the double and triple mutants, where it was unaffected in the cells infected with the deletion to the global regulator MVFR. So it's really a pretty cool observation that they're offering us. And in panel B, they're effectively quantitating the amount of fluorescence that they see. So it basically said, did your eyes judge panel A correctly? And what we see is that your eye was really telling you the truth. Collectively, their results offer that while the intestinal cell lines infected with the knockout mutant MVFR retain staining of this protein, cells infected with wild type or any of the other quorum-sensing mutants had a marked decrease in staining, which meant that the microbe was still able to get across that intestine. And so these findings overall suggested that increased intestinal permeability, and the animal model that they used were mice, may be mediated by disruption of the tight junctions of the intestinal epithelium. In concluding this observational study, they offered us that model that I started us on our journey with, namely that the results suggest that the different effects of what they saw with these quorum-sensing mutants act independently on intestinal inflammation and permeability and are independent of their ability of these regulatory mutants or the wild-type strain to disseminate into the gut. And then they take us down this final path, and if you will, the concluding part of their discussion, which I interpreted as or read as code for their statement offering that while inhibition of this global regulator MVFR through genetic disruption or non-ligand-based inhibitors can prevent this global regulator-driven increases in gut permeabilities. But mutations in LAS-R, which is the common patho-adaptation that has been previously reported for Pseudomonas, may impact the efficacy of a single drug therapy against the global regulator MVFR. So I think more work needs to be done, but it was an absolutely tour de force in genetics and regulation. And I hope it sets up Michelle to talk about her aspect of genetic regulation when we get to the next paper.
You know, Michael, I've always been really impressed and a little bit fearful of Pseudomonas aeruginosa because it's so versatile. In the human body, it can infect skin wounds. It can, I think, affects the eye, cause pneumonia, cause sepsis. So it's clearly very versatile. It's not a, just marching along and doing the same thing no matter where it is. It's got ways to sense what's happening in the environment and then adapt accordingly. So understanding that it has a single quorum sensing, let alone three that are collaborating, goes a long way toward explaining how this simple little gram-negative bacterium can wreak such havoc.
And, I introduce quorum sensing to the medical students, and they say, is this going to really matter in the care of patients? And just thinking about what this one short paper offers us is really remarkable. And as you said, it affects many systems. And oftentimes, we only expose our medical students to one system, one microbe. They don't often learn how versatile these microorganisms are in causing disease in our patients.
Michael, what is the advantage for Pseudomonas to influence barrier permeability? Is there any?
It opens up new niches. What the MVFR global regulator does is it actually makes 60 small molecules, and it's effectively opening up habitat. These 60 small molecules are involved in remodeling biofilms, and that's what Pseudomonas really is designed to do. It's designed to live in dirt and establish itself in a biofilm. I think we're the accidental host in this circumstance. In cystic fibrosis patients, Pseudomonas is looking at the chloride channel defect in those individuals afflicted with that disease, and it's able to set up housekeeping in those individuals' lungs, whereas in you and I, it's ignoring it because it can't set up itself in that niche and establish the alginate biofilms that it needs to really cause substantial pathology in the lungs of CF patients.
Okay.
So Michelle really brought out that this is a versatile, nasty creature that's often pan-resistant to every antibiotic we got in the cupboard.
Yeah. Does it spread readily from one person to another?
No, it's not. It's not typically contagious. You got to get it from the environment.
Yeah, so it's just probably trying to scavenge nutrients and has all these systems in place to degrade whatever polymers are around to liberate nutrients that it needs to build another cell. And it tries desperately when it's in weird sites in the body, but that doesn't necessarily mean it's more fit because it may not spread to another person. But I guess if it spreads to the environment, from a person to the environment, that keeps the cycle going.
Mm-hmm. Yeah, what I think it is, it's not necessarily fitness, it's opening up new niches for it.
Yeah.
That's what the spreading and the gut permeability and inflammation is just bad because once you wake up the immune system, it's like pulling that fire alarm. Those cytokines, as we've all learned from Daniel, are evil.
So this microbe also grows well on plants and in soil, correct? So
yes.
Yeah. So a lot of the selective pressures may well have been in that complex environment that we'll talk about in the next paper.
All right. Thank you, Michael.
You're welcome.
All right, Michelle.
So the title of this paper is Plasmids Manipulate Bacterial Behavior Through Translational Regulatory Crosstalk. And it is the work of a large group. The first author is Catranio Thompson with collaborators Hall, Chandra, Martins, Salbach, Pentorat, Bird, Ford, Little, Piazza, Harrison, Jackson, Brockhurst, and the lab of Jacob Malone. And they are at a number of institutions in England. The main lab is affiliated with the John Ennis Center in Norwich, England, and also the Norwich Research Park. But their collaborators are from the University of Liverpool, University of Sheffield, University of Birmingham, and University of Manchester. And this was published in PLOS Biology last month, February. So they are going to describe a really amazing example of what has been called plasmid chromosome crosstalk, where a regulatory protein encoded on a plasmid can manipulate the expression of the host chromosomal genes. So the host for this particular plasmid is another Pseudomonas fluorescens. And it is also an environmental microbe. It lives around plant roots in the soil, and it alternates between a couple different lifestyles, as Michael was referring to. It can either be motile or it can hunker down and form biofilms. And it is well known that Pseudomonas fluorescens has a signal transduction pathway that can help the organism switch from one lifestyle to another. And what's surprising is one of the key regulators of that switch, developmental switch, is encoded by a little plasmid. So this plasmid, which is called PQBR103, is only 425 kilobases in size, and it can mediate its own transfer from one cell to another through conjugation. It's known to encode resistance to mercury, ultraviolet light. It also can alter more than 400 different bacterial genes encoded by its host, and it encodes a regulatory protein, RSMQ, which is actually broadly conserved in the bacterial world and can control a large number of pathways through a post-transcriptional mechanism that we'll talk more about. But this little plasmid packs a lot of punch. It can, for each nucleotide on the plasmid, it can change more than one host gene. So it's quite a powerful system. So the study began with an in silico search to ask whether this one plasmid is an oddity. Is it the only one that has acquired this regulatory protein RSMQ, or is this more widespread? So they did an in silico analysis looking for other plasmids in the bacterial world that also encode homologs of RSMQ. And they found that 20% of the Pseudomonas taxa have plasmids, or the plasmids carried by Pseudomonas have got a homolog of RSMQ. And more interesting to me is 50% of the Legionella ace taxa, their plasmids encode a homolog of RSMQ. Yet none of the Bacteriaceae plasmids that they looked at have this regulator. So it's clearly widespread, but it's also specialized in particular taxa, and at least the major ones are environmental microbes. So they also learned by doing phylogenetic analysis that the plasmids that encode these RSM homologs are themselves diverse. So it didn't come from a single evolutionary event, but rather there have been apparently multiple transfers from host chromosome onto plasmids, and then the plasmids have themselves diversified independently. So it looks as though there's strong selective pressure on the plasmid biology to retain one of these, and that it confers fitness. So having convinced themselves that this is likely to have a large impact in the microbial world, they were interested in whether the RSM function that has been well described in Pseudomonas, Legionella, E. coli, many other microbes, whether the plasmid encoded gene retains those functions. So they first asked whether the RSMQ protein is like the bacterial homologs and able to bind mRNAs and block or activate their translation. So it's well known that these RSM proteins can bind to the nucleosome or ribosome binding site on an mRNA and affect translation. So they turned to a high throughput method that they're going to use throughout this paper, which is a version of surface plasmid resonance, a highly sensitive technique where you have a substrate that you can coat with a target molecule, and then you bind your protein of interest to it, and then you can flow different candidates across that chip. And if they bind, then when you shine a beam of electromagnetic power onto that chip, if there's a protein bound, then the beam will bounce off in a different manner that can be quantified. So it's a way to quickly assess and quantify binding to a particular substrate. And in this case, they are going to use single-strand DNA because RSM is known to bind RNA. So this is a good surrogate for that. So what they first...
That is so cool.
Yeah.
That is so cool.
They used it very effectively, too. And I did a quick search to learn more about this particular technique, which is the version is called Reusable DNA Capture Technology, REDCAT. And there are a number of commercial entities out there that will analyze your samples for a fee. So it's an accessible technology as well. So they first did a search and found, in fact, that their RSM homolog could bind to the known motifs on mRNAs that the chromosomal homologs bind to. And in and then by doing some site-directed mutagenesis, they verified that it binds in the ribosome binding site of those RNAs or near it. They then went on to ask whether it can interact with other components of this well-described pathway. And these include, actually, before I get to that, they then asked whether expression of this plasmid-encoded RSM actually altered the RNA profile of the bacteria that harbored this plasmid. So they first did RNA-seq to look at the RNA profiles, and they didn't see much of a difference. But they weren't discouraged because they knew well from the literature that this acts at a post-transcriptional mechanism by altering translation of those mRNAs. So they then went on and compared the proteome profiles of Pseudomonas that were carrying plasmids that either did or did not encode this RSMQ protein. And there they saw really impressive differences. And they are illustrated in nice pie charts in Figure 3, where they were especially struck by the capacity of RSMQ that's encoded by this plasmid to suppress the expression of a number of genes that are involved in amino acid and carbohydrate transport and also metabolism. Meanwhile, they would the regulator increases the expression of genes that contribute to motility, chemotaxis, and also remodeling of the cell wall and the envelope. So that's a clear sign that the plasmid-encoded RSMQ homolog is able to alter the developmental switch between am I in growth mode or am I in spread and join a biofilm type mode. So having established that the protein in vitro could bind to these ribosome binding sites on mRNA, they then asked whether it interacted with other known components of the canonical signal transduction pathway. So in Pseudomonas and other bacterial species, it's known that there's a two-component regulatory system called GAC-AS that can sense external signals and then through a phospholrelay turn on expression of two non-coding RNAs called RSMY and RSMZ. These non-coding RNAs physically interact with the RSMQ protein or its homologs and therefore compete with its binding to mRNAs and essentially pull it away from the mRNA to interfere with the RSM's either induction of translation or inhibition of translation. So again, they went to this surface plasmid resonance chip assay, and they verified that indeed the RSM protein encoded by the plasmid could bind to these non-coding regulatory RNAs, RSMY and RSMZ. So it's also known, so that indicated one mechanism by which this plasmid RSMQ could alter the biology of its host by physically interacting with the host-encoded RSMY and Z non-coding RNA regulatory factors. But they also know from the literature that the RSM proteins function as dimers. So they were curious whether the plasmid-encoded RSMQ could interfere with the host biology by physically pushing aside one of the host RSMs and forming heterodimers with the host RSM proteins. So again, they went to a really lovely assay that relies on an old favorite, beta-gal assay. So this is a two-hybrid system assay that they did in E. coli that takes advantage of an assay where two different subunits or domains of the adenylate cyclase enzyme are encoded on different molecules. You can then make a fusion protein and put your regulatory protein of interest, in this case RSMQ, and then a target protein, a host RSM homolog, onto different domains of the adenylate cyclase, and then put those into the E. coli. And if the two RSM subunits form a stable heterodimer in this case, then the adenylate cyclase will generate cyclic AMP, and you can detect that in a colorimetric assay on agar. So that is shown in figure 5D, And what was really impressive is that this plasmid-encoded RSMQ in this E. coli assay can certainly form heterodimers with 3 different RSM homologs encoded by the host Pseudomonas. So that would be another way that this plasmid-encoded protein could interfere or modulate the host regulatory system. They then started to look at what the impact was on cells. So rather than do these really beautiful quantitative biochemical assays in vitro, they expressed or studied the phenotypes of Pseudomonas that either encoded or lacked this RSMQ protein. So in figure 6a, we can look at really lovely swarming motility assays. So Pseudomonas fluorescens on particular agar will swim out and form these really beautiful structures, which are quite easy to judge. They found that if they overexpress the RSMQ on a high copy plasmid, induce its high expression, they could completely repress this swarming motility. Now, things are a little more complicated than that, they realized, because when they instead looked at expression of RSMQ by Pseudomonas when it's in its natural state on its native promoter, native plasmid, the phenotypes were more mild or not apparent at all. So this got them thinking more about environmental cues and what could be regulating expression. And they were, of course, aware that the natural environment for Pseudomonas fluorescens is on soil, plants, and roots. And they, of course, had their proteomic data, which had indicated that one of the impacts of RSMQ expression on Pseudomonas fluorescens was on utilization of particular carbohydrates, amino acids, etc. So they did some of these, decided to look at conjugation. Recall, this is a plasmid that can be transferred by conjugation. And they did those assays on different growth media. And what they found is when they cultured Pseudomonas on a minimal agar, they saw that the RSMQ protein increased efficiency of its own transfer to a neighboring cell via conjugation. But they didn't see that when the cells were grown on rich media. So again, consistent with this recurring theme that they're seeing in their data, that RSMQ is really responding to the nutrient conditions, and its activity is dictated by that. So they looked in a little more detail using a biolog screen. So biolog is another company that provides terrific resources for us. You can purchase from biolog multi-well plates where each well has got a different component. So they purchased plates that had different carbon sources, for example, and then screened strains of Pseudomonas that encoded the plasmid RSMQ or not. And what they found is, yes, indeed, that the expression of RSMQ was affecting the ability of Pseudomonas fluorescens to metabolize different carbon sources. They have a lot more that they can pursue to try to understand that in detail, but it was clear from both their molecular data and also these more bacteriological assays with whole cells that RSMQ on the plasmid was clearly responsive to local nutrient cues and was dictating traits such as, do I turn on my chemotaxis machinery? Do I turn on my flagellar apparatus and become highly modal? Or instead, do I switch to a mode where I'm really focused on acquiring amino acids and carbohydrates? So how might this relate to the biology of the plasmid, for example? It makes really beautiful sense if you think about it. If the plasmid is sitting in a host cell that is surrounded by ample nutrients, if that plasmid stays put, it's going to efficiently spread to all the progeny of its host. Therefore, it's spreading efficiently vertically by just staying put. But if it finds itself in a cell where the nutrients are limiting within the host, it perhaps is a better strategy to exit and start spreading by horizontal gene transfer. So in other words, manipulating the cell translation machinery so that it shuts off nutrient acquisition machinery and instead turns on machinery that can promote cell-cell interaction, including chemotaxis, motility, production of extracellular polysaccharide matrix to mediate cell-cell interactions that can then make conjugation efficiently, and the plasmid then would spread to neighbors. So
when it's hungry, it goes on a mission to find food. And that's effectively what this regulatory transcriptional regulator is actually doing. It's, I'm hungry, I got to go find some food.
Or there's no food here, so there's no point in my sticking around because you're clearly not multiplying. So I'll just spread over to the next cell and the next cell and the next cell.
We've all had those roommates who would raid other people's food out of the fridge. So
what was striking to me about this, besides just how impressive the really logical molecular and biochemical quantitative assays they did to demonstrate that this RSMQ homolog was really acting and interacting with each of the levels of the this classic signal transduction pathway, the GAC-AS, RSM-YZ, RSM pathway to control translation, but also that it's really a plasmid strategy toxin, let's say, that's really quite distinct from maybe the classics that we think about, like a pore-forming toxin that lyses a red blood cell to liberate, hemoglobin, to release iron for the bacterium. Instead, it's really getting into the heart of the developmental pathways and the metabolic pathways and dictating whether the host cell is in a sessile mode or a modal, let's go join a biofilm or interact with other cell modes. So that's what I have to share. And I think, Vincent, you were able to learn more about the first author.
Yeah, so this is Catriona, I guess that's how you say it, who is, by the way, a really good field hockey player.
All right. I was a field hockey player, Catriona.
Yeah, I don't know. A lot of scientists should be that. So Catriona's career path has been a little unconventional. She fell into microbiology during her first PhD year, did her first rotation in cancer biology, and she thought, yeah, that's where I'm going to go, to cancer biology. But then she did a project in the labs of Professors Walker and Byron looking at protein antibiotics and said, this is what I want to do. She also got interested in plant health and how microbes impact that. So she ended up doing a postdoc with Jake Malone at the John Innes Center, looking at plasmid-encoded regulators and how they impact bacterial phenotypes. And from that work, she was able to create and mold this project, which is the PLOS Bio paper we've just heard, and find her niche. We've been talking about niches today. She found it. And she wants to thank, besides people in the Malone lab, Rob Jackson, Michael Brockhurst, Ellie Harrison, and Jamie Hall, who helped steer the project and were great sounding boards for ideas and always supportive. And how about an exciting day? Well, she says, all my days are exciting, so I can't pick a typical one. But when she looked at her SPR data, this was a new technique that had been developed on the back of the ARD-CAT system at JIC, and they didn't know if it was going to work, and she saw that it did, and she was happy about that, and other people have been using it ever since then. And for advice to young colleagues, have a healthy work-life balance.
Yay.
She said during her PhD, she had periods where I worked insane numbers of hours, and once she stopped doing that, she became a better scientist. She also found that the times when she's most fulfilled in the job are when she could take a step back from it. And she said it's important to spend time outside the lab to recharge, gather thoughts, and help see the bigger picture. And so, Kat, thank you for those perspectives and recommendations, and keep it up.
And I'm feeling some kinship with Kat, although I've never met her. In addition to also being a field hockey player and a believer in work-life balance, two students from my lab have been studied a integrative conjugative element, a mobile element of Legionella, and it is a large family that's spread throughout the genus, and each of those integrative conjugative elements encode a homologue of RSM, which we in the Legionella side call Caesar T. So by doing bioinformatic analysis, Zach Abbott and Caitlin Flynn in my lab also found that the RSM homologues dictated whether the Legionella was in a swim mode or a replication mode. And it also has a conjugation system affiliated with it. So it's another example of the paradigm that they've established here of these RSMs being encoded on mobile horizontal elements and altering the biology of its host to further their own interests, which is to make more of themselves.
That's what Stan Falco used to say, right?
Yeah,
that's right.
The goal of every microbe is to make 2 microbes. And I guess the same is true of a plasmid or a integrative conjugative element.
Goal of every virus is to make a lot of viruses.
Yeah. That's because they're so limited in their genomes.
Well, they don't divide, right? They just build new ones. And you say if you're going to build one, you might as well build 1,000,000. So that's what they do. Thank you, Michelle. I have a couple of emails I want to share with you. One is from Carl. Dear TWiM team, thanks for a fascinating episode discussing the gut microbiome and its possible link to depression. That was episode 278, Bacteria Sing the Blues. I had to laugh at the very end of the episode when you discussed Merv Zeden's habit of cold water swimming in the Atlantic Ocean during the winter. Michael Schmidt posited that the Gulf Stream current perhaps keeps the waters off Galway warm during the winter. I can assure you this is not the case. I heard this episode late as I had been away in Galway during New Year's week on a family vacation. On a bus tour of Galway, we saw swimmers near Black Rock Diving Tower. My future son-in-law, probably, decided he must do this. With my whole family watching, he plunged into the 7-degree sea water, 44.6 degrees F. He quickly learned about the physiology of flirting with cold water shock.
Oof.
While he told us about how exhilarating the swim was, the critical care doc in me was concerned about hypothermia. We got him all warmed up without incident. So in this study of N = 1, we conclude that the Gulf Stream hypothesis does not hold water.
Hold water.
Bump. Thank you, Carl.
Thanks for all you do. Carl is a DO in Middletown, New Jersey. And one is from Henley, who writes, I'll confess, I'm more of a regular TWiV listener than TWiM, but I happened to listen to episode 281. Excellent overall. I think there was some conflation around energy from fusion versus from the fuel process being discussed. It felt like a very apples and oranges comparison. Biofuels, like this is the one where bacteria make fuel, right?
Right.
Yeah. Biofuels are really a repackaging of energy. Energy must come from somewhere, and for plant-related endeavors, it's usually from solar, which is ultimately fusion, or some other conventional source, if it's one of those indoor vertical farms with artificial light. So the fuel discussed on the show is not really a new form of energy. It's just another way to store it, an awesomely dense way. Fusion is interesting to study since it potentially allows us to directly tap a source of energy that can add to the portfolio of humanity's energy sources. I do wonder about bang for buck in bio versus fusion study, but I suspect there's a higher minimum investment required to do anything experimental in the latter. The mind-shifting realization when you start thinking about the biofuel process is that plants are actually pretty inefficient when it comes to gathering and storing solar energy that we can use relative to modern solar panels. And she gives some calculations that I'll go over with you in a moment. For extra fun, consider that humans use energy at a rate of about 100 watts based on a 2K calorie diet, and that one modern 1.6 square meter solar panel generates about 300 plus watts under full sun. Makes one wonder where super efficient food calorie production could go in the long run. Thanks for all you do, Henley. All right, so here are some rough notes around using using values plucked and roughly cross-checked across from the web. Let's assume that we're not converting to ethanol. We get all the food calories. This ignores potentially using the cob and other byproducts. So 4184 joules per food calorie times 860 food calories per kilogram of corn times 25.4 kilograms per bushel equals 91 megajoules per bushel. And
that's a lot of joules.
And that is 7.7 times 10 to the 9th joules per acre per year. An estimate for power production of 1 acre of solar panels, 300 megawatts per acre acre. Yeah, a low end estimate is 1 times 10 to the 12 joules per acre per year. So there's about 100-fold difference, which is roughly in line with some of the ethanol production versus solar discussions I see floating around. So the solar is way better, right?
100 times. Yeah, 100 times better.
That sun is just great stuff. You should appreciate it on a nice sunny day.
Yeah.
That's right.
But, I appreciate...
Today was.
I appreciate their clarification of the energy transduction versus energy creation. I think Petra was trying to clarify that for me too. She was also a little bit skeptical that this was going to be sufficiently efficient, the biofuel that we talked about.
Too bad.
But, this is the sort of discussion that we need to be having with once that undergrad walks into their introduction to science. Yeah. They need to be thinking about these things because this is where the new ideas will come. And let's face it, we're going to need a lot of new ideas as we deal with things like global climate change and how to feed people. I mean, he's absolutely right. No one wants to eat electrons. I mean, we haven't figured out how to eat electricity yet. We want to eat that steak and that corn on the cob and, that great salad. We don't want to eat a light bulb.
So if you make the electrons tasty, Michael, and you'll eat them.
That's true. Well, the only thing that I know that can make electrons taste tasty are the replicators on Star Trek. Yeah. And we haven't figured out how to do that, where they convert energy into matter.
Yeah. Well,
we have to go 3 more centuries into the future.
And that's a different podcast as far as I'm concerned.
That's a different podcast. That is indeed.
And this podcast is TWiM, and that is 283. Show notes, microbe.tv/TWiM. If you want to send in a question or a comment, we love to get them. We really do. TWiM@microbe.tv. And if you like what we do, consider supporting us. We run on your support. We don't use ads on our programs to keep them pure. So we need your support. And whatever you can supply, $1 a month would be great. Less than a cup of coffee. $1 a month, it's nothing. And if you folks all did $1 a month, we could do a lot of cool things. So how do you do that? Microbe.tv/contribute. There are a couple of ways you can do that. Michelle Swanson is at the University of Michigan. Thank you, Michelle.
Thank you.
And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.